Thanks so much, everyone, for staying the distance on today. I joined at 9 o'clock or whenever you kicked off with this kind of carnival atmosphere from Arvind and colleagues, and I'm amazed that you're still here to listen to me speak about this. Thanks to Michelle for the story about Katie, and I think, the key messages from this day are probably the patient voices that you're hearing, which are so much more powerful than people like me waffling on. But you've had an introduction to this condition, nephronophthisis, and I'm going to talk to you very briefly in the time available about some of the science underlying this and present another case just to re-emphasize the sort of clinical presentations of this condition. I'm an adult nephrologist from Newcastle, but I have the privilege of doing clinics with my pediatric colleagues, both Sally Johnson and Yinsent Say, where we run a renal genetics clinic, and also we run a specialist tuberous sclerosis clinic, actually. But within the multidisciplinary renal genetics clinic, we see families either with strange diagnoses like nephronophthisis or families without a diagnosis, and try and offer them something in terms of a diagnosis and management. We're talking about genetics of kidney disease here, and just to remind you that you've become pediatric nephrologists, and your bread and butter is inherited kidney diseases. 70% of all pediatric nephrology is said to be genetic, whereas only 10% of adult kidney disease is perhaps genetic. But there are growing numbers of genes involved in kidney failure, and in fact GFR is estimated to be in the heritability of GFR is supposed to be 30 to 60%. And something like a third of all adults with end-stage kidney disease will report a positive family history of kidney disease. And so, in this MDT environment, I'm enjoying seeing families with these disorders. Rodney mentioned the primary cilium. The cilium is this antennae that extends from the cell surface. And I've got a picture of a cartoon and a picture of one here. So the cilium is said to have a 9+0 structure if it's a primary cilium, and a 9+2 structure if it's a motile cilium. We don't need to know too much about these, but essentially the cilium is like a flagpole extending from the apical surface, and it tells the cell what's top and what's bottom, what's left and what's right, and feeds back information from extracellular clues back to the nucleus to allow certain genetic programs to occur. In the cilium, in the tubules, the cilium project into the lumen, and so here you can see electron micrographs of renal tubules with the cilium poking out into the lumen. And these cilium are decorated with lots of proteins that sense the environment. They have a mechanosensor role. They have an osmosensor role. They have a signaling role. So literally these cilium are wafting around in the urine, determining the flow of urine and determining literally the taste of the urine or the osmolytes in the urine, and feeding that information back to the cells around them. So quite a fascinating organelle. And as Rodney alluded to, every gene that encodes a protein that's expressed in the primary cilium, if that gene goes wrong, it tends to give a cystic kidney disease phenotype. So the paradigm is that if you have a gene such as polycystin 1 and polycystin 2 that's defective, it gives cystic kidney disease, probably in part due to its role in the primary cilium. We've heard about the huge spectrum of nephronophthisis. So nephronophthisis is this kidney disorder that leads to kidney disruption. It's autosomal recessive in the vast majority of cases that we've heard. But I often irritate people by saying it's a cystic kidney disease. Actually, the cysts don't have to be there and can be a late feature. And really, the size of the kidneys is small rather than large, like a PKD kidney. And it's a renal fibrotic condition as well as a sort of cystic kidney disease. The cysts probably come about from dilatation of tubules due to atrophy of surrounding tubules. And this sort of Greek term of disappearance of nephrons literally describes what is seen under the microscope. The extrarenal manifestations that we've heard about, such as loss of vision, skeletal abnormalities, liver fibrosis, occur in about 10 to 20% of cases, and those will point you towards a ciliopathy syndrome. So if you see kidney failure or chronic kidney disease in the context of a sort of syndromic presentation with eye, brain, skeletal, liver abnormalities, then you really should be thinking about a ciliopathy. And I don't need to go through all this, but there's a huge collection of syndromes like Joubert syndrome, BBS, Jeune syndrome that are part of the ciliopathy spectrum of conditions. So how might it present? So children typically present with polyuria and polydipsia due to a loss of urinary concentration ability. And in some children, they may present with complications of CKD, such as anemia and poor growth. Typically on urine dipstick, you might not see any blood or protein, so the urine is bland and doesn't give you any clues. There may be secondary enuresis, so the child may be typically dry at night and then start bedwetting again. As Rodney alluded to, sometimes there is a sort of early onset presentation with cystic kidney disease that looks like ARPKD, and certain genetic forms of nephronophthisis, typically MPHP2 or inversin MPHP3, CEP83 and NEC8, have all been associated with this very antenatal or perinatal presentation of enlarged cystic kidney disease that just looks like ARPKD and may fool you. And the urine, sort of lack of urine concentration ability, I think is a key clue. So I see lots of families with Joubert syndrome with brain and eye involvement, and the families want to know, is my kid going to get kidney disease? And so one of the early sort of screening tools that we have for this set of patients is to look for loss of urinary concentration ability. And the kidney ultrasound may be useful in determining the echo texture of the kidney and for cyst development. Biopsies, we don't traditionally biopsy this set of patients anymore because the genetics has allowed us a diagnostic route, but his historical biopsies show, this scarring of the kidney, fibrosis of the glomeruli and the tubules. Periglomerular fibrosis is shown by this white arrow here. Sclerosed glomeruli is shown below. And typically it's a sort of global, non-specific pattern of, what you'd associate with end-stage kidney disease. If you do EM, you see tubular basement membrane disruption with thickening and thinning and multi-layering of the basement membranes along the tubule. Again, a non-specific but significant finding in end-stage kidney disease. And so people have asked the question, well, is this confined to pediatrics, or do adults have nephronophthisis as well? And so this was an interesting study done by Nini Neurs, who's a geneticist in the Netherlands, and Albertine van Erd. And they took an end-stage population all been transplanted and said, how many of these have got an MPHP1 gene deletion? And actually they found the incidence was something like 0.5%, so a significant amount of undiagnosed dialysis and transplant patients had an MPHP1 deletion. So it does present later in life in adults, and typically we would label these adults as end stage of unknown cause. Either they might be biopsied that showed non-specific findings or were never biopsied at all. The genetic spectrum is huge, and so I think there's now 18 causes of MPH, but if you look at ciliopathies, there's maybe 90 or more genes involved in ciliopathies, and this is growing every time. But MPHP1 remains the most common genetic diagnosis, accounting for around a quarter. Of those MPHP1 diagnoses, half of them will have the whole gene deletion syndrome, so the MPHP1 gene is deleted. And there still remains a number of cases that are unsolved, and that keeps people like me busy. And so if you go onto OMIM, you can list the 18 causes of MPHP. So this is small print stuff. You can draw diagrams, and this is out of date now, but we used to call nephronophthisis infantile, juvenile, adolescent. But I think, this is outdated, and we talk about nephronophthisis in terms of gene identification. So it'd be nephronophthisis type 1 for NPHP1 and nephronophthisis type 2 for inversin, etc. And as Rodney alluded to, the most common genetic form of the infantile presentation now is NPHP3. So NPHP2 remains incredibly rare. It's associated with situs inversus and various other skeletal abnormalities. But NPHP3 can cause a renal limited, very early onset, severe cystic kidney disease phenotype that ends in end-stage renal failure very early in life. Okay. I'm going to skip that. So how do we think about genetics? So people have done gene panels, exomes, genomes. What's the best thing to do in investigating these people? So various people have published, largely from the Friedhelm Hildebrandt lab that I spent some time in a few years ago. But if you do an exome of children with increased echogenicity of their kidneys and suspect nephronophthisis, you solve over half the cases. But actually, when they looked at these cases, actually a third of them had completely different diagnosis to nephronophthisis. So it just shows you that end-stage kidney disease is a variable, phenotype with lots of causes. If you go to the Genomics England and say, can I do a ciliopathies panel? They'll say yes, if the patient fits the criteria. And that ciliopathy panel will allow 73 genes to be screened that of various causes of renal ciliopathies of different flavors. If you're just in a resource limited country, then you can simply look for NPHP1, which is the most commonest hit by PCR tests, and you can identify whole gene deletions very easily and very cheaply. But if that fails, then you might progress to a panel or an exome or a genome. And I think the way we're moving in the UK is that we will end up applying a whole genome sequence and then a virtual panel to diagnose these cases. And people have shown that whole genome actually does pick up stuff that you would miss with panels, and deep intronic alleles have been found for various of these NPHP genes so far. And as Michelle mentioned, me and my colleague, Dr. Shrivastava in Sunderland, added NPHP to the ARPKD radar group. So now if you've got any patients with nephronophthisis, you can add them to radar. And certainly there are active Facebook groups for patients with nephronophthisis. And I think patients are encouraged by knowing that there's someone else out there with these rare diagnoses, and they get some encouragement by talking to each other in that way. There's a Joubert syndrome UK group that I'm part of, and many of these patients do have nephronophthisis as part of their Joubert syndrome. Briefly, I'll just tell you about this case. So this is a young girl that presented in Newcastle, and she was 12, presented with abdominal pain, and screening bloods at that time showed a very marked reduction in kidney function. The kidneys were bright but normal size. The urine was bland. There was no family history of renal disease. The child had some autistic tendencies. The child was biopsied, and it showed chronic tube interstitial damage that I've described, consistent with nephronophthisis. And this family went into the 100,000 Genomes project for whole genome sequencing. Mom and dad and the affected here went into that project. But as typical sort of Genomics England style, they reported no mutations found, and it was kind of shelved for a while. And I was asked to review the case. And unfortunately, as the spelling of nephronophthisis is difficult, this family had been recruited under this panel of nephrolithiasis rather than nephronophthisis. So they'd had a kidney stone gene panel applied to their data, and no one had even looked at the NPHP1 genes. So we opened the panel up and applied an NPHP panel. Actually, that came out to be negative as well, which shows the challenges of this. In the meantime, quite sadly, her older brother, who'd just finished his A-levels, crash landed to adult nephrology, requiring dialysis. And at this point, with 2 affected family members, I thought this has to be genetic. And basically, I manually curated the raw data and looked at what's called the BAM file, which is is the calls of the data. And you don't really need to understand this, but here in these tiny gray dots are the individual reads. So you can see hundreds of reads here and hundreds of reads here, but across this region, which is just fresh air essentially, this is where MPHB1 lies. So these two children had a complete whole gene deletion of MPHB1 with their raw data showing no reads of MPHB1 in this area. And so, when the data was queried, like, is there a mutation in MPHB1? They said no, but there wasn't, they said no because there wasn't even any calls in MPHB1. And so this is a weakness of some of the analysis. And what's quite tragic for this family is that there's a third sibling who also carries the homozygous gene deletion, who's at present has normal kidney function, but this family is obviously sitting on a time bomb with the kidney function. So there are a few learning points from this case, I think, that, phenotypically characterizing patients is vital, and recording the information is vital, and spelling nephronophthisis is vital. The biopsy may or may not be helpful, but applying certainly the right set of gene tests to the right phenotype is crucial. Consider the evolving clinical picture in relatives, particularly siblings and maybe cousins. If the gene panel is negative, widen the search. And, consider weird things that might not be picked up on panels, like whole gene deletion syndromes. Okay, so I think I'm towards the end of my time, so I'm just going to recap what I told you, or tried to tell you. Nephronophthisis presents insidiously with really polydipsia, polyuria, maybe some growth problems and anemia. Cysts aren't diagnostic and maybe only seen in a few patients, maybe up to half. Hypertension is not a strong phenotype, actually, but they can present with hypertension, but typically the salt wasting protects against hypertension. And genetic testing is the best way to diagnose nephronophthisis. And I think kidney biopsy should be a historic investigation for these cases, given the power of molecular genetics. Okay, so I'll stop there and we'll open up for some discussion. I think Arvin might have some sort of special questions to ask you, actually.
Ooh. I was going to ask, yes, we can do this. Let's see if they've been listening, John. I have a sneaky suspicion we won't get the responses we want, but let's have a listen, shall we? Let's try. One of the things I was going to say while we're waiting for the Slido to go up, John, is what about one of the things that we chatted about was, what do you tend to find on urine dipstick? I don't know whether people are listening to Rodney, they may or may not have switched off. So it's always just one of these things I always think, oh, perhaps it's something else.
It's usually bland, the urine dipstick. If there's severe hypertension, you might see a little bit of protein, but it's never nephrotic range proteinuria. And, as Michelle has talked about her daughter, transplantation is curative for this condition. It doesn't come back in the transplanted kidney. So, that's a very positive sign. In terms of therapies, we don't have specific therapies for MPHP. We have good blood pressure control with ACE inhibitors, but we don't have a therapy that switches off this condition yet. But obviously that's what we're working at here in Newcastle.
Okay. We're going to now poll your questions, you'll be pleased to hear. Sorry.
Fiendish questions coming up on the poll, John, but can I just extend your answer to the previous question? When these patients present in adult life, they are, as you say, they often don't have much in the way of cysts, and they have a rather bland urine, and actually they look like interstitial kidney disease. And there is, of course, now this whole family of autosomal dominant interstitial kidney disease. These often aren't, or usually aren't, autosomal dominant, but they're the same clinical presentation, many of them. And I think that's something we're missing out on in our current descriptive accounts.
Yeah, so I think there is a phenotypic overlap with, as you say, with mucin 1 and neuromodulin and some of the newer ones. But family history is key, and so the autosomal dominant pattern should give it away. And certainly UMOD and MUC1 are fully penetrant, so you would find a a positive family history of renal failure. With nephronophthisis, the parents will be unaffected. They'll have normal kidney scans, normal kidney function. Clearly, you need to be thinking about consanguinity if there's, to help you make a recessive diagnosis as well. So, these are key questions when you're asking a family history.
Okay, let's get on to flip to the Slido poll, which at the moment is showing up the most common genetic cause of nephronophthisis is CEP290. True or false?
Neil, are you able to see the results?
Yeah.
88% say false.
88% say it's not. Yeah.
Oh, well, I'll keep quiet.
So MPHP1 would be the most common cause.
Yeah.
Next question.
Is there another question?
There's three. He gave three, so we will not insult the man and not play them.
Infantile nephronophthisis is commonly seen with NPHP3. True or false? 73% are with true. Oh, it's falling. It's 2-1 in favor of true.
The answer is true, yes. It's the most common genetic cause of infantile nephronophthisis, MPHP3.
Wow.
So this it should no longer be thought of as adolescent nephronophthisis. That's a load of nonsense now.
Some of the figures you're coming up with for population surveys are making it clear that it's actually not quite such a rare disease as we used to call it either, if they're representative. Whole gene deletions of NPHP1 are unusual, true or false? And after the answer to this one, John, you can tell us whether that was that algorithm has been fixed. Because I'm now worrying about that in the negative returns I've had from the Human Genome Project.
So, of the 25% of patients that have MPHP1 deletions, around a quarter of them will have a whole gene deletion, either homozygously or compound heterozygosly with a missense or something else.
Well, I'm completely with the audience in that case who aren't sure whether a quarter of them, it makes it usual or unusual.
Yeah.
It's not usual, but not rare. Okay. The audience is equally divided on that one, John.
Yeah. I think it's just to be aware that, whole gene deletions, you can get caught out with these simple panel tests that just tell you if a mutation's been seen or not, and they might not do a gene dosage assay.
Yeah, that's important. There's two genetic questions being posed on Slido as well. So they're the opposite screen, so the opposite extreme. So one is, I struggle to get genetic testing for unexplained CKD, bland urine with strong family history. And the other one is, I do get whole genome sequencing, but it comes back full of variants of uncertain significance.
Yeah, so I think we have to work with our clinical genetics colleagues is the answer. And as I mentioned, I do at this MDT clinic. I don't propose to understand it all myself, but, certainly as we sequence more and more patients, these so-called variants of uncertain significance get reclassified, actually. So PKD1 is probably the most common gene we all sequence. Yeah. And typically a VUS will turn into a variant of likely pathogenicity when more families are established with the same allele. And so this will happen for all forms of genetic kidney disease in the future. So, your genomics medicine hub should be able to provide you with a gene test for unexplained kidney failure, and you'll be able to get the data. And, the beauty of the genomic sequencing is that you can ask it for it to be reanalyzed at a later date when more information is available on different genes.
So are they getting good now at doing those reanalyses and getting back to the clinician and patient?
They're getting good at listening to me moan at them, but so I told them about they're missing the MPHP1 deletions, and we did a analysis and found I think 6 other families that they've missed. And it doesn't sound a lot, but for those families, a diagnosis is vital. Yeah. And so, they've wised up to this fact that they were missing this point. I think that my point of mislabeling the phenotype from nephrolithiasis to nephronophthisis was unique. That was just a medical student entering the wrong thing in a pilot study patient. So hopefully that's not happening for other cases, but it just shows you that these schoolboy errors could occur.
Yeah, it needs to be unknown CKD-ciliopathy or something instead, doesn't it? John, we're past 5 o'clock. You did fantastically well in getting what is a really complex disease clear in such a short space of time, and keeping it really exciting on where it's going and the genetics. And another time, maybe talk about therapeutic options.